Rufino. I am a second-year biology major at Foothill College, and the project that we were working on, me and my mentor, is binding and structural analysis of neutralizing Fabs in complex with coronavirus spike protein peptides. And this is an image at the lab where we were working at. So what is the big picture with our project? We want to be able to understand the mechanisms of, mechanisms of the neutralizing antibodies against coronavirus. And this can potentially help combat COVID-19 and possible future coronavirus outbreaks. This is important because most people have gone through a pandemic and many people were impacted by COVID-19. The focus of our research can be split into three goals. So our first goal was to investigate how these antibodies bind to the virus proteins. Our second goal is to visualize the binding process. And our third goal is to build 3D models so that we can analyze them. So at the bottom, I have images of an antibody. That V-shaped section is called the Fab region. And that is the part that is responsible for binding to the coronavirus spike protein. So on the very right, we have a picture of a coronavirus, and the little structures that stick out of it are the spike proteins. And in that middle image, those red sections are what's called fusion peptides, which is where the antibody can bind to in order to neutralize it. So how do we get to that point? For our first goal, we want to see how well the antibody Fabs bind to the fusion peptides. So to do this, we perform what's called a bilayer interferometry. And this is essentially an experiment that allows us to measure how strong the connection is between our two proteins, so our antibody Fabs and our fusion peptides. From there, we go into crystallography. So we will then get these complexes of our proteins, combine them and put them into solutions that crystallize. We will collect these crystals, send them out for data collection. And after rounds of data collection, we then get to use this data with another set of data processing software to build a 3D model. So for our first results, we had 12 complexes that we were working with. Unfortunately, due to a lack of resources, we had to focus with only 3. These 3 complexes use human coronavirus 229E, which is associated with colds, and SARS-CoV-2, which is COVID-19 virus. So in our graphs, we see increasing values, which imply association, and we see decreasing values, which imply dissociation. So the proteins attaching and detaching from each other. Using this as a reference, we then get to work with crystallography. So with these 3 complexes that we made, we set out crystal trays in different crystallizing solutions, and we monitor the crystals growing over a period of time of days, and we select crystals that we believe are good for data collection. Usually bigger crystals are better. And we take note of the solutions so that we can prep them properly for to send them out to be processed. The way we process them, first we have to collect them using a little tool. It's a loop that is kind of like a spoon. It's microscopic level, so it requires a lot of precision. We scoop out the crystals, we prepare them, we send them out, and using some X-ray imaging, we then get images that look like this. And using a data collecting software, it'll look for patterns in these images. So like, for example, those little spots, it'll collect data for us. And using this data that we collect, we then get to put it into another data processing software, which will generate a map for us to build in. Those green areas is where the computer wants us to put stuff in, and the red is where we don't. And the blue area is where is actually a reference model we use from someone else to add on to using our new data. And those ribbony shapes are the fusion peptides of our antibodies, and that green area is what we believe to be coronavirus peptide. So this is a good sign for us. And as we build, we can also use references from other researchers' models to compare with our own. So on the left is the reference model, and on the right is my model. We can layer them on top of each other so that we can compare them and see where we need to change, what we need to add, and get an idea of everything that we're building as well. So in conclusion, in summer, we were able to confirm binding with our fusion peptides of the coronavirus along with the antibodies and form crystal complexes for model building. Then we started building these models, and all of this is could be used potentially to help develop treatment for coronaviruses. Some next steps that include building and refining. We had a bunch of crystals that we didn't actually get to building models for. And then something else, I had mentioned the lack of resources for the other complexes that we left out. So in the future, it's something we can look into. And for me, finishing building the model that I displayed because it is currently incomplete. Some skills that I learned are important lab skills like performing bilayer interferometry, crystallography, and data collection, building and refinement. Some challenges including troubleshooting due to resources, timing with equipment and availability, and little mistakes that were made here and there. Some successes include the amount of progress we've made given the limited amount of time we had for this internship. And some key takeaways, it exposed me to the world of research. I was able to develop a lot of lab skills for the future, and it gave me a lot more confidence in STEM. And lastly, I want to give some big thank yous to the MESA team for telling me about the opportunity for the internship, SLI staff and Barnes Lab for the internship opportunity itself, Foothill College and SLI for the scholarship funding, my mentor, Sheena Vasquez, who helped guide me along the way. Honestly, I couldn't have asked for a better mentor. And the staff at Barnes Lab and Stanford for welcoming me into their space and helping out with the research. Thank you.